Welcome back, Bio30 Prep. We are going to continue our discussion of the excretory system by looking at urine formation and the function of the nephron. In your textbook, it's pages 311 to 315. By the end of this lecture, you will be able to describe the function of the nephron and explain how it removes waste from blood plasma, and you'll be able to describe the process of urine formation by the nephron. Now, when we look at the nephron, we're going to use an analogy to explain how the nephron works. The cleaning of the blood requires the removal of wastes, excess salt, water. It can be compared to house cleaning. If you think about cleaning out a desk drawer that has been around for a while, lots of pens and pencils, but also a lot of junk just sitting in that drawer, there's two ways to clean it out. The first method is to dump everything out onto the floor and then go back and pick up the useful items. The other way to do it, leaving behind a pile of garbage that you can then just scoop up and throw away. Another method would be to sort through the drawer, picking out all the garbage and leaving only the useful items behind. Now, for those of you who have done any house cleaning, I think you can agree with me that the first method would be faster, and that's the way the nephron works as well. What's going to happen is all the stuff in the blood, except for cells and a few big proteins, are going to get dumped out of the circulatory system into the nephron. Then, the useful molecules are going to be reclaimed back into the blood, leaving the waste behind to be excreted. And so, if you want to go back, take a look at blood composition just to see, or just to get a better understanding of how it's cleansed. So, urine formation is a four-stage process. The first step is glomerular filtration, and this is where the filtrate is removed from the blood. It's the dumping of the drawer. It's the movement of fluids and molecules and ions from the blood into Bowman's capsule. Tubular reabsorption is taking that filtrate and returning the good stuff back to the circulatory system. So ions and water are going to be transported into the blood from the nephron. The transport of ions, some of it's going to be active transport, some of it's going to be diffusion, and for water, most of the water is going to move back by osmosis. The next step is when some of that, or when the useful stuff, when the good stuff is being taken back into the blood, some waste material follows with the osmosis of water. So tubular secretion is a double check. It's going through the good stuff and making sure that there isn't waste, and taking out some of that waste material that made it back into the drawer or back into the blood. So it's going to add waste to the filtrate from the blood, or take waste from the circulatory system and put it back into the nephron. After reabsorption, the blood sends some waste back to the nephron through active transport. Because we've got to dig through to find the waste, this is going to require ATP. It's going to require energy to get all that waste out of the blood. Finally, water reabsorption. It's going to be happening throughout the entire process, but at the very end, any excess water is going to be returned to the blood from the filtrate. We want to preserve as much water as possible. So the first thing we're going to look at is blood flow. Blood comes from the renal artery, and the renal artery branches into a bunch of arterioles, afferent arterioles. And if we just take a look at blood composition in the afferent arteriole, it's the same as normal blood, as blood in the rest of the body. It's going to contain your blood cells, your red and white blood cells and platelets, proteins, protons, bicarbonate ions, sodium ions, potassium ions, chloride ions, glucose, amino acids, and waste material. That blood gets pumped into the glomerulus, which is that capillary bed that by pressure, a bunch of stuff is going to get forced out into Bowman's capsule. If we take a look at the proximal tubule, so here's Bowman's capsule, and from Bowman's capsule we enter the proximal tubule. If we take a look at what's inside the proximal tubule, it contains protons, which are hydrogen ions, bicarbonate ions, sodium ions, potassium ions, chloride ions, glucose, amino acid, and waste, or basically all of the small molecules that are normally in blood get forced out into the proximal tubule. The part of the circulatory system that leaves the glomerulus, the efferent arteriole, or the outgoing arteriole, contains only cells and proteins, so it's deficient in a ton of stuff. So in the nephron, these slender tubules are the functional unit of the kidney. There's about 1 million in each kidney. The renal artery, as I said, branches into many afferent arterioles, each of which goes into a glomerulus, and then from the glomerulus, this is the site of filtration. All of those small molecules are removed from the blood, forced out by blood pressure into Bowman's capsule. Note, we're not deoxygenating the blood. If we take a look at our diagram, the blood vessel going into the glomerulus and the blood vessel coming out of the glomerulus still contain oxygen, red blood, blood cells that are rich in oxygen. The capillaries join to form the efferent arteriole, and the efferent arteriole, notice that it comes back and it comes over and it surrounds the rest of the nephron. And this is because all of that good stuff that got dumped into our nephron has to be reabsorbed back into our circulatory system. So the blood's not deoxygenated, and that's because the hemoglobin, which is a protein in the red blood cells, doesn't deoxygenate. The capillaries, because blood is deficient in most substances, the efferent arteriole then passes a net around the collection tubule, and the collection tubule, both the proximal and distal, and this is where we're going to have reabsorption, or much of the filtrate is reabsorbed back into the blood vessel. So again, filtration, it's a high pressure system in the glomerulus. Urine is formed by blood pressure forcing the plasma portion of our blood into the nephron. In the kidneys, about 180 liters of fluid passes into Bowman's capsule every day, or into the million Bowman's capsules that fill our two kidneys. 179 liters are returned back to the circulatory system. So most of that fluid does come back into our circulatory system by reabsorption. In the filtration, here are the substances that are filtrated out of our blood: water, salts, sugars, amino acids, hydrogen ions, other small molecules. They're all found in Bowman's capsules. This is all called filtrate. It also includes the waste. So the filtrate is what enters Bowman's capsule and the proximal tubule. The blood cells and blood proteins remain inside the circulatory system. Now we have to do some reabsorption. If we take a look at the diagram here, they've highlighted the first part of the proximal tubule. This highlight can really be anywhere along here. They've simplified the diagram a little bit just to make it a little easier to read. But all along that proximal tubule, reabsorption is going to happen. Without reabsorption, we would have to urinate every 5 minutes, and we would have to drink a liter of water about every 10 minutes if we didn't reabsorb that water back into our circulatory system. Over 99% of the filtrate is going to be returned to the efferent arteriole. And reabsorption is selective. The first things that are reabsorbed, sodium, it's actively transported from the nephron, so it requires ATP. And the negative ions are going to follow that positively charged sodium ion by static electric attraction. The maximum amount of material that can be reabsorbed is termed the threshold level. If we have excess amounts of material, it will remain in the filtrate and become part of our urine. The movement of these ions creates osmotic pressure, causing water to flow from the nephron back into the blood. And what's happening, if we take a look at the little picture here we got our transport mechanisms, and they're color-coded: active transport, passive transport, and osmosis. Solutes are active and passively transported. The active one is sodium. The passive one would be your chloride ions, and your bicarbonate ions just by electric attraction. Sodiums are moving across, they're positively charged, so the negatively charged ions will follow. Water is going to follow because we're going to end up with a high concentration of ions in our blood because they're being actively transported in. So the filtrate is going to end up with a low concentration of ions. And so here we have a concentration gradient. Water is going to try and balance it, so osmosis carries the water out, trying to make the concentrations equal. Now, because water is moving across, some wastes are also carried across as well. So, but for the most part, waste is staying behind in the as filtrate in the nephron. There's reabsorption of ions and osmosis of water back into the circulatory system. Now, the loop of Henle is a special part of the nephron. It's going to aid in the reabsorption of water and ions from the nephron to the blood. Now, when we took a look at our kidney, we gave it two regions, and I'll just draw a little kidney over here. We had a cortex, which was the outside of the kidney, and the medulla, which was the inside of the kidney, and they were different colors. And it turns out that the medulla has a higher salt concentration than the cortex. What this means is, as the nephron passes from the cortex into the medulla, it enters a saltier region, which is going to allow for the more osmosis of water. So the descending loop, that's the loop that's going down. So if we take a look, the filtrate is flowing this way through our nephron, through the loop, and then back up the distal tubule. And the loop separates the two, so the proximal tubule is on this side. The distal tubule is on the other side of the loop of Henle. And the loop of Henle is this narrow part of the nephron where the tube decreases in size. So the descending loop is permeable to water and has a low permeability to ions. So what happens is we have high salt concentration in the medulla, low salt concentration in the nephron, and water is going to get pulled by osmosis from the nephron to the circulatory system. So that's the reabsorption of water. As we go back up the tubule, permeability changes. It's going to be highly permeable to ions and not permeable to water. So now, on the ascending loop, we're going to have the movement of sodium ions by active transport, chloride ions, and bicarbonate ions as they follow the sodiums. And so they're going to diffuse into the circulatory system and into the medulla, maintaining that high sodium concentration, that high salt concentration. As we go back up, and as we follow our tubule around, it joins a collecting duct. And the collecting duct, again, passes through that salty medulla, and what's going to happen is now most of our ions are out, our nutrients are out, all that's left is waste, uric acid and urea, and water. And so what's going to happen is, as we enter this medulla again, water leaves the collecting duct. And so we're always trying to maintain water, keep as much water inside of our body as possible, so that we don't have to keep taking water in to have our body function properly. So there's also secretion from the distal tubule, and this is moving waste from the circulatory system back out into our nephron. So after reabsorption of waste material, the excess potassium ions and the excess hydrogen ions, these are moved by active transport back into the nephron. Also, molecules that aren't normally a part of the body are also secreted from the distal tubule, things like antibiotics. Like reabsorption, this is the active transport of molecules. The concentration of waste after reabsorption is low, so in order to get that waste out of our circulatory system, it has to be actively transported, which means it requires the use of ATP. The energy for secretion comes from cells loaded with mitochondria located around the nephron. So outside the nephron are tons of cells that have lots of mitochondria. They're breaking down or metabolizing sugar to get those ATP molecules in order to actively transport waste from our circulatory system. More reabsorption from the collecting duct. Water is the main chemical reabsorbed. Everything else is pretty much, if it's a necessary chemical, has already been reabsorbed back into the circulatory system. All that's left is the water. This is going to increase the concentration of our urine. Again, it occurs by osmosis. The collecting duct, as we said, it passes through that salty medullary tissue of the kidney. Water is going to be drawn out of the collecting duct into the kidney and then into our circulatory system. And the amount reabsorbed is actually controlled by hormones that affect the permeability of the collecting duct, and those hormones we're going to look at in the next lecture. So in summary, from this lecture you should get the idea that most of the constituents of blood plasma are forced into Bowman's capsule at the glomerulus by filtration. The major exception are the blood proteins. Also blood cells stay behind in our circulatory system. The nutrients and other useful substances, such as sodium ions, are returned to the capillaries surrounding the proximal tubule by active transport, diffusion, or electric attraction. Water is reabsorbed throughout the process by osmosis, especially in the descending loop of Henle and the collecting duct. The ascending loop of Henle is primarily for ion reabsorption. And then there's going to be secretion of, by active transport, of excess materials or waste materials that are still in the blood. So for example, excess potassium ions and hydrogen ions are lost from the body in this way. The waste in the collecting ducts is going to enter the renal pelvis, or all those collecting ducts in each kidney or from each nephron are going to merge into the renal pelvis, and the renal pelvis is going to transport that material, that waste product, to the bladder through the ureters. In our next lecture, we're going to look at some of those regulating factors that control how much water is reabsorbed, how many ions are reabsorbed back into the circulatory system.